Donc on va commencer, je pense que beaucoup d'entre vous connaissent Guillaume Salbreux, qui a fait donc sa thèse avec nous à Curie, qui a été pendant longtemps à Dresde, après à Londres, et qui depuis un an ou peut-être un petit peu plus est à l'Université de Genève maintenant, et il va nous parler donc de Connecting Scales in Tissue Morphogenesis. Vas-y Guillaume.
Okay, thank you, Jean-François. Thanks a lot for the invitation to speak. It's a pleasure. Yeah, I'm going to talk about tissue morphogenesis and about the question of connecting different scales. And I'm going to start with something that is going to be obvious to physicists, which is that in physics, we are often interested in connecting scales and being able to go from the microscopic scales of interacting molecules to the macroscopic scales of thermodynamic variables, such as the concentration, the pressure. And at this macroscopic scale, we describe the system by fields which represent quantities which are averaged over many macroscopic molecules. And one very simple example of this is to consider the motion of the fluid inside the pipe, a very simple problem. And we know that in such a case, there is a quasi profile that establishes itself inside the pipe. And the velocity of the flow is given by a simple relation in which there are some geometrical parameters such characterizing the size of the tube. The velocity also depends on the pressure that is applied. And there is only one parameter that really describes what the fluid is doing, and that is the viscosity. And so this tells us two things. One is that to describe the system at the macroscopic scale, it is enough to know only one parameter, and there is a very generic behavior that doesn't depend on the microscopic properties of the fluid, of the emerging flow profile inside the system. And the other thing we learn is that if we want to characterize how a fluid is going to flow, it's enough to ask what is this viscosity and how can the viscosity of the fluid be related to what happens at the microscopic level. And the reason I'm mentioning this is because we are facing similar questions in biology. And notably, in the morphogenesis of tissues, we have to connect different scales. And this is a beautiful movie from the lab of Susan Eaton, which is showing the morphogenesis of a Drosophila pupal wing. And so initially the tissue is roughly rectangular. It has, and then it progressively deforms and adopts the shape of an actual wing. And at the end, one can recognize what is going to be roughly the shape of the adult wing. One also sees that as time progresses, there is a pattern appearing on the surface of the wing, and that's the patterns of the veins which establish itself over time. And so what's really nice about this movie is that we can see simultaneously the macroscopic flow, the macroscopic shape change of the tissue, but we can also see every individual cells, and we can in principle track every cell and follow it and know what it's doing, when it's dividing, when it's changing neighbors. And we have thousands of these cells. And so we see that the question of morphogenesis, one question about morphogenesis, is to be able to relate what the cells are doing at the cellular level to the macroscopic tissue flow or tissue patterning that we observe. And I would argue that if we think about what's going on in this movie, there are two essential things. One is that there is a mechanical aspect to it, which is that every cell is exerting forces. These forces are transmitted from cell to cell, and in the end, the macroscopic stress profile inside the tissue is creating the deformation and the flow that we observe. At the same time, there is also signaling going on, notably in the emergence of this vein pattern that was visible in the movie. And the mechanism behind these signaling processes is that some cells secrete molecules, morphogens, which then diffuse in the extracellular space, bind to receptors of other cells, and then there is information processing that other cells can do depending on the morphogens that they sense. And then they can react to it and take a decision, for instance, becoming a vein cell or not becoming a vein cell. And so in both aspects, in the mechanical aspect and the signaling aspect, there is a question of connecting scales from molecules to tissues. And the physical framework, and notably the framework of active matter physics, can help us with this, and it is relevant to understand biology for several reasons. One is that in a physical system, one is often considering assemblies, collective effects of many individual objects, for instance, the collective motion of active particles. And similarly, in a tissue, there are many molecules in the cell or many cells in the tissue. There is also the fact that in active matter physics, the individual particles are out of equilibrium at the molecular level. For instance, if one consider heated genus particle, which create the fact that they are heated create a temperature gradient, resulting in a self-thermophoresis of particle and their spontaneous motion. And here the energy is provided by a laser that hits the particles. And in a cell, the fact that the cell uses nutrients and oxygen to produce ATP brings it out of equilibrium, as then ATP hydrolysis, the energy of ATP hydrolysis can be used by the cell to power its internal processes. And one last thing is that in active matter physics, one often looks at broken symmetry variables in active liquid crystals. And this also occurs in a biological system because there are cytoskeletal filaments which are polarized inside the cell, but also at the scale of the tissue because individual cells are polarized because there are a set of proteins that can segregate inside a single cell and then establish a cellular axis, or because simply the cellular shapes themselves are elongated. And this also introduces some level of order. And one very nice example of this connection, which is actually from the work of Jean-François and Pascal Sebelzan, is this picture here, which shows cells that are cultured in vitro in a circular dish. And the cells turned out to have an elongated shape. And it also turns out that they tend to be aligned along the circumference of the disc. And because of this, they have to, and also they tend to elongate with a relatively coherent pattern on long time scales. And the fact that there is this boundary condition on the alignment of the cells at the boundary of the disk imposes a topological charge inside the disk. So there must be defects. And in fact, there are two defects, 2 + 1/2 lemmatic defects that emerge on long time scale inside the system. And what's nice is that it's actually possible to predict that there are only going to be 2 defects and also their position inside the disk based on a simple theory which takes into account the cost of distortion in the elongation field. And this simple theory predicts what is observed experimentally. And so there is a nice connection here between ideas of soft matter physics and active matter physics and the behavior of a biological system whose large complexity ends up being summarized in the simplified description where there is elongation field and the effective energy of this elongation field. So the question I would like to discuss today are other examples maybe of how this relation can be made. And I'm going to talk about mechanics and signaling. And so in the first part, I'm going to discuss mechanics and how cells, cellular forces turn into tissue deformation in three-dimensional morphogenesis. And in the second part, I'm going to talk about signaling and morphogen gradients, and particularly synthetic morphogen gradients, and how to relate the molecular properties of morphogen gradients to tissue scale behavior. So about the tissue morphogenesis, I will first describe a little bit experiments that have been done by our collaborators at the Crick Institute in the group of Axel Behrens. And this group is interested in, they're studying cancer biology, and they are interested in notably about the pancreatic cancers. And they use mice as a model stem to study the evolution of these cancers. And so those are images of the pancreas of the mouse. The left image shows the entire pancreas, and the right image shows the close-up view, in which one can see that the pancreas is actually made of a tree of tubes. And as one goes deeper into the tree, this is what the right schematic shows, the diameter of the tube gets smaller and smaller. Okay, so the pancreas is essentially a tree of epithelial tubes. And then to study the cancer growth, they use tamoxifen injections to induce tumors inside the pancreas. And they do this by having genetically modified mice which react to tamoxifen injection, so that some cells randomly undergo activation of an oncogene and deletion of a tumor suppressor gene. And then these random cells start to proliferate much faster than the pancreatic cells would do normally. And after a few weeks, the single cell have transformed into a small clone, so early lesion that contains a few cells inside the pancreas. And then they have developed a method to be able to image at the cellular resolution what the pancreas looks like after the tumor injection. And so in this movie, one can see one of these pancreatic tubes, and the red color indicates the cancerous cells. And then one can see that they tend to locally deform and form small bulges inside the pancreas. And due to this imaging, one can really quantify the shapes that these tumors have. And if one does this, one can see that the tumor lesion, the cancerous lesion, can be intratubular or extratubular. So this 3D view and this cross-section is showing an intratubular tumor. So in the cross-section, the yellow outline, the yellow line is showing the outline of the tube, and the red cells are a tumor which has grown towards the inside. And one can also observe the opposite deformation. One can see that in some places, the tumor locally enlarges the size of the tube and grows towards the outside. And then when they looked at where the tumors were growing, they saw that their position depends on the diameter of the tube. And this is what this graph is quantifying. If the tube have a small diameter, then they tend to make tumors that grow to the outside. Whereas if the tubes have a large diameter, they make tumors that tend to go to the inside. Okay, and so that's what we wanted to understand. And so we turned to the question of what is the mechanics of this epithelium. And so the tissue that lines the pancreas is made of cells that are is made of an epithelium, a single cell epithelium, so of cells that are attached to each other. Each cell has a polarity, so it can distinguish the top from the down, the apical side from the basal side. And the filaments inside and inside the cell, there are cytoskeletal filaments and motors which exert forces, which result in internal tensions, which then are transmitted from cell to cell and generate tensions inside the epithelium. And to illustrate this, I'm showing images from the different model system from Drosophila. But this movie shows the surface of a tissue during Drosophila development. And the nice thing about this movie is that one can see the myosin in green and the outline of the cells in red. Because the myosin is pulsatile in this movie, one can see that every time that the myosin concentration intensifies, there is a constriction occurring. And so one can visualize how this myosin molecular motor on the cellular scale are generating contractile deformations. And another classic experiment that shows the existence of these internal contractile forces is the laser ablation, which is done here in the junction in the wing disc. And if one does this, one can see that there is a recoil occurring of the junction, and so that reveals the internal tension that exists inside the tissue. So then we saw, okay, we should then look at the distribution of myosin molecular motors inside the tissue, and more specifically, we look at phosphomyosin light chain, so which marks active myosin inside the tissue. And one can look in a region around the tumor and compare, and then the top image here shows shows the myosin level, and the lower image shows in red where are the transformed cancerous cells. And then we take an axis along the apico-basal line of each cell and quantify the distribution of myosin. And so the myosin is largely either basal or apical. And if we look at the profile, we see that in the wild-type tissue, there is a lot more myosin apically than basally. But the distribution is modified in the transformed cells, and the levels of phosphomyosin light chain become more equal to each other. So there is a perturbation of the distribution of motors inside the cancerous cells. And so what that tells us, it gives us a picture for the force distribution inside this tube. So this is a schematic of the cross-section of the tube. The apical side of the cell is facing to the inside, and the basal side is facing the outside. And based on the myosin light chain distribution, because there is more myosin apically, we would also expect that there is a higher tension apically than basally. But this is perturbed in the transformed cells. The two tensions based on myosin level become more equal to each other. And so then the question is, what is the effect of, so there are two things that happen in the tumor, the cells proliferate, and there is also this redistribution of myosin molecular motors. And the question is, what does it do? And so to answer this, we have used a 3D vertex model that we have developed, where the distribution of cells in the tissue are represented by apical network and a basal network of vertices, which are points which are located at tricellular junction. So they represent the point where several cells meet. And so here we have the apical network, the basal network, and we can define which vertices enclose which cell inside the tissue. And then the vertices can go anywhere in 3 dimensions so that the tissue can adopt its preferred or its mechanically balanced shape in 3 dimensions. So that defines the geometry of the tissue, but we also need to define the forces. And so in order to capture forces that occur in the tissue, we write a differential virtual work function, which allows to connect cellular forces to forces acting on vertices. And so this differential virtual work function captures what we think is essential about the cellular mechanics in the tissue. And so the first term in this work describes as a pressure. The pressure is coupled to a change of volume. And in our case, we assume that the pressure acts to maintain a constant cell volume, because experimentally, if we measure the cell volume of transformed cells and or wild-type cells, we see that it's the same. So it looks like cells maintain a fixed volume. In addition, there can be a surface tension on different cellular interfaces, which is associated to changes in cell areas, in surface areas. And there can also be a line tension because it's possible that there are specific structure along the cellular interfaces, such as actin junctional cable, which exert a tension specifically on junctions, like we were seeing in this laser ablation movie of the wing disc. And then from this virtual work function, one can calculate the force acting on the vertices simply by taking the ratio of the virtual function and the displacement, infinitesimal displacement of a vertex. And then we evolve the shape by assuming that the vertices move according to these forces. Okay, and so then we with this model we generate different tubes with different radii. So we adjust our parameters so that the shape of the cell, the aspect ratio of cells is like the one we measure in experiments. And we also generate tubes of different size simply by changing the number of cells around the contour of the tube, because we have measured this experimentally, and we see that the tube is not bigger because the cells are larger. The tube is bigger because there are more cells, and the cells have the same size roughly, no matter the size of the pancreas. So, okay, and then starting from this initial condition, one can take a cell inside the tube and then assume that and then say that it is transformed, which means for us that it changes its forces. It has a different basal and apical tension, And also it starts to proliferate and undergo several rounds of cell division. Then we run the simulation and observe the outcome. And so the left is a 3D view, the middle image is a cross-section, and the right image is an experimental cross-section. And without further effort, we observe the same thing as in experiment, which is that in a small tube, the tumors grow to the outside. And in a large tube, they invaginate and tend to go towards the inside. Because this is what is seen notably in the cross-section, the function of the tube diameter. Okay, so qualitatively, this fits what we see, and quantitatively, we measured the deformation of the tumors depending on the size of the duct diameter. So this is what we see in the left schematic for experiments, where positive value corresponds to deformation of the tumor away from the tube, and negative value corresponds to deformation towards the center of the tube. And the blue points and red points are taken at 2 different times after the initiation of the tumor. And the right plot corresponds to the same quantification, but done in simulation, when the time actually corresponds to a number of divisions. If we do this, we see that we have the same trends, and also the duct diameter, the tube diameter at which the behavior changes from going to the outside to going to the inside is the same in experiment and in simulation. And in addition, we also wanted to test. So in the model, we do 2 things. We have cell proliferation, and we also have changes in surface tension of apical and basal surface tension. And to test which one was more important, we also have done simulation with just cell division, so just over proliferation. And if we do this, this quantification is showing that then the tumor never grow to the inside. There is always an enlargement of the tubes, the simulated tumors, and they always grow to the outside. It's actually possible to do the same thing in experiments, because it turns out that if one just deletes the tumor suppressor gene, the cancerous cells over-proliferate, but there is no change in myosin distribution. And in that case, we also observed that they always grow to the outside. So this behavior is dependent on the redistribution of myosin inside the cells. And so to understand this a bit better than running simulations, it's actually helpful to move from a description at the cellular level to go to description as a surface, a continuum theory that allows to understand more clearly what is going on. And so we have developed a theory with coworkers. We have developed a theory of active surfaces, where the idea is that we represent a tissue now by a single surface, but the real tissue has a thickness. And so across the cross-section of the tissue, there are stresses, and the sum of these stresses give rise to a tension, and differential stresses that act along the cross-section can give rise to a torque, so a torque acting along the cross-section, which tends to induce rotations of the surface. And so this kind of idea has been used for a long time in elastic shells. And here we have adapted it to describe an active surface where there are internal tensions and torque which depend on flows, on normal or tangential flows in the surface, and also on active processes. And specifically in the context of a tissue, we think that we know that the cytoskeleton is present apically and basally, and exerts different surface tension apically and basally. And so if we think of an epithelium as a bilayer with 2 surfaces under 2 different tensions and a given height, and then we move to this simplified continuum surface theory, the tension of the layer is the sum of the apical and basal tension, and the local torque that is induced depends on the difference between the apical and basal tension. So differences in apical and basal tension creates internal active torques. And then so we can write a version of this active surface theory, which we think is appropriate for epithelial sheets. where there is a tension which contains an elastic term in blue, describing the resistance to change in area, and an active tension. And there is a resistance to bending with a bending modulus, which contributes a term to the internal torques, as well as an active bending moment. So the underlying hypothesis of this model is that tissues are elastic with respect to area changes. So cells have a preferred area. And so if we stretch uniformly, there are elastic stresses building up. But they are fluid with respect to shear flows. And this is because we know that cells can rearrange with topological transition and can relax shear stresses that would emerge in the system. We also assume that tissues can resist bending and have a preferred curvature. And so these differences of apical and basal tension can create internal active bending moments. Okay, and if we now consider an application of this theory in a very simple setup, where we start from a planar sheet and then we allow for deformation away from the planar state with deformation UN, we can write down an equation. The left term of these equations, when the tension is positive, are all trying to keep the surface flat. So, and the resistance comes from the bending modulus, from the internal tension, or from possible attachment of the tissue to a substrate. And the right term is driving deformation, and it's driven by gradients of the active bending moment. And an application of this here is to simply solve the equation for a line with two regions, a red region which is active, let's say, which is under high bending moments, and a blue region which is passive, and the gradients of active bending moments then give rise to a fold. And so this is in line with the usual observation in epithelial tissue that the formation of folds is associated to an increase in apical contractility, which in this framework is creating an active bending moment and generates a deformation. And now we return to the question of the tumor growth in the ducts. We can now apply this continuum theory by taking a cylinder and we assume that there is a region of the tumor which is growing, but is also subjected to an active torque. And then if we solve for the deformation of this region, we see that it depends on the radius, and overall, the deformation of the center of the tumor can be to the outside or the inside. And there is a threshold radius at which the behavior changes, which depends on the bending modulus and the difference of active torque. Okay, and then if we look in the equation, we can see also where this behavior comes from. And essentially, there is a competition between evagination due to the growth, because the cell proliferation gives rise to a bigger tumor, and therefore a local enlargement of the size of the tube. So that favors growth to the outside. But at the same time, the redistribution of forces of myosin to the apical and basal side creates an active torque, which leads to a spontaneous curving, which tends to bend the tissue towards the inside. And if we had an infinite cylinder, then it would always go to the inside, because the first effect becomes negligible, because the increase of the tumor size relative to the overall tube size would become negligible. So this is why at large, at least in the theory, this is why at large radii, the tumors tend to go to the inside, but not at small radii. And interestingly, one can also look at tumors that are grown in other tissues, in the liver and in the lung, and there is a roughly consistent tube diameter of tubes, at which also in its other organs, one can see that above the radius, the tumors are growing inside, and below the radius, they are growing outside. So with this in mind, so with the idea that this surface theory and active torques can capture some aspect of epithelial deformation, we have also done something a bit more exploratory and looked at deformations of pattern epithelia. So the idea here is that in the recent years, there are many examples of observation of organoids, which are structures grown in vitro from stem cells. And depending on the medium, the stem cells are driven to differentiate into different types. And they can then form complicated structure, which recapitulates some aspects of the actual organ. And so the self-organizing process that is observed here is also very interesting from a physical perspective. And it's been argued that often the growth of these organoids starts by the formation of an isotropic shell with a lumen inside the tissue. And then there is a symmetry breaking event, possibly due to signaling, which creates a polarity inside the system, and cells change their behavior and change their type in different regions. And then one can ask whether this initial symmetry breaking later leads to patterning of forces inside the tissue, which would create the first deformations, which are the origin of the growth of the organ. And so here we investigate this from a theoretical perspective with our model of epithelia, with the idea that we simply take a sphere and we assume that it's patterned, and that now there are two different types, blue cell type and a red cell types, which can exert different forces. And if we assume that there is just a pattern of tension, so the red active region is under higher tension than the blue region, with our hypothesis on how epithelia behave, we will not see a three-dimensional deformation because the active tension constricts until the tension is uniformly balanced in the sphere and there is no three-dimensional deformation. So instead, we looked into what happens when you have different levels of active torques in different parts of the tissue. And then we ask what three-dimensional deformation one can expect. And so we consider axially a symmetric shell, and we start from a sphere, and we have an active region with a non-zero internal active torque and a passive region with a zero active torque, and here the volume inside the sphere is fixed. And then one can calculate the shape, and this movie here is showing a sequence of steady state shapes which are obtained by progressively increasing the active torque in the active region. And in order to classify a little bit what these shapes look like, we can build a phase diagram where on the x-axis we have the magnitude of the active torque in the active region, which increases. So the left is a sphere, and then as one moves to the right, the deformations increase. And on the y-axis, we have the size of the active domain. And what we see here is that we essentially, in the bottom and top of the diagram, we get budding, which is something that is observed in actual organoids. This is seen here, for instance, in images of growth of the intestinal organoids. And then on the left of the diagram, there is we don't have solutions in here, and this is both, there is self-intersection, either because there is a formation of the ideal neck, infinitesimal neck that connects two spheres, a sphere that is either to the outside or to the inside. And in the middle region of the diagram, there is a bit of a strange behavior with folding on itself on the surface. So here we have generated this phase diagram, but we were also we observed buds, but not a lot more than this. And so we thought that to generate the diversity of shapes that are observed in these organoids, we should maybe add more ingredients. And so we have considered also nematic spheres. And because we assume axis symmetry, there are two defects at the poles. And we assume that the nematic field is described by a passive effective energy, which contains the Landau-Deugen energy and the Frank free energy, which penalizes distortion. And then now we assume that our internal active torques, they are driven by this nematic field, which means that we have oriented internal stresses, which are not the same epically and basally. And then we change our constitutive equation for internal active torques by adding also a nematic term where the torque is dependent on the internal order in the surface. And that sounds a bit far-fetched maybe, but actually one can, that there should be internal talks that are oriented by nematic order. But one can find biological examples, and notably there's been a very recent work by the group of Keren looking at alignment of fibers in hydra. And the images are showing that the surface of the hydra is made of two tissues which contain fibers which are oriented perpendicular to each other, which is indeed what would be needed, for instance, to generate an internal active torque oriented along a nematic field. And in the right, there's a schematic showing what they observed, which is that, interestingly, the nematic field forms defects at specific locations in the animal, in the mouth and in the foot, and also at the tip of a tentacle. There is a defect which is balanced by 2 - H defect at the bottom of the tentacle. Okay, and so then we calculate our shapes now that we have this internal active torque oriented by a nematic field. And depending on the sign of the contractile forces, if we start from a uniformly patterned sphere, we can generate cylinders. So the sphere progressively elongates, and the length of the cylinder increases linearly with the magnitude of the nematic torque. And conversely, if we change the sign of the nematic torque, then we progressively move to a torus, which ends with a self-intersection. So there is a progressive constriction of the center of the sphere, and at the last point is a self-intersection, which, if there was a topological transition, would lead to a torus. And if we mix now an active and a passive region, we get either the growth of appendages. So the active region starts to form a tube that emerges from the sphere. Or we can also grow this tube, but towards the inside of the sphere, and there is a self-intersection line where the appendage that is growing is reaching the other side of the sphere. And this is reminiscent of what is seen in sea urchin gastrulation, where the mesoderm invaginates towards the inside of the embryo, and that leads to that is the gastrulation event in the sea urchin. And so here we are trying to simply explore what shapes we can obtain with these ingredients, and we have a sort of shape zoology, where if we have isotropic active torque, we can get budding or this strange curling behavior, or we can also grow tubes, an appendage or torus. And so now it would be interesting to see if in a biological system we can observe these nematic fields and if they are the origin of some of the deformation that can be observed in these organoids. So as I mentioned initially, I also wanted to talk a little bit about signaling, and I'm going to talk about synthetic morphogen gradients. And this is something that we have done in collaboration with the group of Jean-Paul Vincent in the creek. And so this is a project about morphogens. And to start with a little bit of history, the morphogens the term was coined by Alan Turing in 1952 in his famous paper on the chemical basis of morphogenesis, and he called a morphogen a substance which can be a form producer. And so after this, there's been many studies of the actual morphogens that exist in biological system, from which there is a relatively simple picture that emerged, which is that you can get a morphogen gradient by having a source. And so some cells are special, source cells, they produce the morphogen. And then the morphogens diffuse inside the tissue. And at the same time, it is degraded. And this behavior allows to build up an exponentially decreasing concentration profile away from the source with the characteristic length scale, which is given by the ratio of the diffusion constant and the degradation rate. And so the idea is that then the different level of concentration can be read by the cell to get positional information and know what their distance is relative to the source. So that's a nice and understandable picture. But one outstanding question is what exactly happens if we move away from this macroscopic picture and look a little bit more closely inside the tissue. And there things are not so obvious because the morphogen needs to diffuse into the intercellular space. They probably also interact with receptors on the cell surface. And the behavior and the diffusion and the degradation of morphogen will be influenced by this environment. And so there has been different proposals on mechanisms that could affect this. One of them is the bucket brigade. So the idea that morphogens could undergo directed transport on receptors. Another in transcytosis, the idea that a morphogen could be endocytosed inside the cell and then exocytosed somewhere else, which would contribute to its motion inside the tissue. And another idea is hindered diffusion and the possibility that a morphogen would bind to extracellular fibers, which then slows down its progression inside the tissue. And so one of the models that has been used a lot to look at morphogen gradients is the DPP Drosophila wing disc morphogen gradients. This is a schematic of these gradients. There is indeed a DPP production stripe in the center of the tissue from which DPP is produced. And then DPP diffuses away inside the tissue, creates a gradient. And this gradient is thought to instruct then the position of the veins on the adult Drosophila wing, the same vein pattern that we were seeing in the movie earlier. And maybe I should mention that the schematic here on the left is showing an earlier stage than the pupil wing movie I was showing before. Okay, and DPP is essential for this. And in fact, if there is no DPP, not only we don't see veins, but also there is also a problem with growth. The tissue is not growing properly. So then the idea of Christina and Jean-Paul has been to take a wing, Drosophila disc, where DPP has been removed. And instead, they've been doing something really nice, which is that they have genetically modified the fly so that inside the source cells, inside the source region, the cells start to produce GFP, so fluorescent protein, which is inert, in principle, does not interact with other components of the tissue. And then they also have modified the cells in the posterior compartments so that they express the receptor, which has been engineered to have a nanobody which binds specifically to GFP. So they have artificially recreated with genetic transformation the simple picture of formation of the morphogen gradients with secretion at the source and binding to receptors inside the tissue. And so what's nice is that now there is a lot more control than one would normally have on what the gradient is doing, because there's no hidden effect. In principle, the GFP is not interacting with other receptors than the one that have been put artificially. And so if we look at now at a wing with no receptor, so we see the source because the intracellular GFP is visible. And outside we see nothing because there are no receptors expressed, and probably the extracellular GFP is washed away before the imaging is done. But then if one puts a high affinity receptor in the compartment, now we can see a gradient. So we see the GFP signal, and we see that indeed it's decaying progressively away from the source as one would expect. And so then the question is, how does it compare to the perfect picture of an exponentially decreasing gradient? And it turns out that it's slightly different from what we expect. So this is now a measure of the GFP intensity away from the source inside the tissue. And in the middle, there is a decreasing exponential, as we expected. But then in the left, there is a shoulder. And in the right, there is a non-zero tail. So the GFP level does not decay to zero at large distance, as one would expect for a normal morphogen. So to understand where this comes from, we have done a slightly more detailed model of the diffusion of the GFP, which is here our artificial morphogen, where we have simple diffusion in the extracellular space. But we also have, so C is the concentration of GFP, D is the diffusion constant. And then we also have binding and unbinding to the artificially engineered receptors with the nice thing that we know the on and off rate because they have been measured independently. And we also include the possibility that the GFP moves away from the tissue into the hemolymph, which is the fluid inside the Drosophila larvae in which the wing disc is sitting, because there is no reason a priori for which the GFP should not exit the tissue. In principle, if it's free to diffuse, it should also be free to diffuse inside the hemolymph. And if we compare the volume of the hemolymph to the total volume of the producing disc, its magnitude is not so large. So it's possible that GFP accumulates in the hemolymph. And then we also take into account the internalization and degradation of receptor in the cell and degradation of ligands in the hemolymph. Now, to measure a bit more what these parameters are, we have done a few additional experiments where we use a GFP bus to see how GFP behaves when it interacts with the tissue. And so one simple experiment is to put the disc in contact with the GFP bus, wait for GFP to go inside, remove the bus, and then wait for GFP to be degraded. And that gives us an idea of how often the receptors are internalized and degraded. And then if we look at the measurement of GFP intensity as a function of time, we see that degradation is quite slow. It's on the so the GFP intensity decreases slowly. So degradation of receptor is larger than a few hours. We have also done a slightly more subtle experiment where we put the disc in contact with the GFP bus for 3 minutes, and then we stop and we look at how the GFP has invaded the tissue along the apicobasal axis. And we do this with different concentration of bus, and then we obtain these images, and we see that there is an apicobasal gradient of GFP building up. We can use our model, because it's the same model also, but in the perpendicular direction, to see if we can predict these gradients. And if we now compare the predicted gradients, which are the bottom graph, to the measured gradients along the apicobasal direction, we can extract some of our parameters, and notably the number, the density of receptor, and we find that there are about 8,000 receptors per cell. Okay, and then if we put this together, we can compare theory profile to experimental profile, and we can explain the shape of the measured morphogen profile, where near the source we have a shoulder because of saturation, because the concentration of GFP is high enough near the source that all the receptors are being occupied, and one cannot go above the maximum concentration where all receptors are occupied. We find that there is a plateau at very large distance, which is actually due to leakage of GFP in the hemolymph, which then comes back and establishes a uniform background inside the tissue. And to test this, it's possible to do an experiment where one expresses in the fat body another organ, receptors that then absorb the GFP. And if we do this experiment, the plateau due to leakage in the hemolymph disappears. So that really shows that this background level is due to hemolymph to the GFP flow diffusing into the hemolymph. We can also change the properties of the receptors. One can use a receptor which has a lower affinity for GFP, and then we get a basal flat level because GFP diffuses into the hemolymph, but the tissue captures only little of the GFP, and because of this the combination of these two effects, we have a low flat level. One can also increase the affinity of the tissue for the morphogen by by increasing the density of receptor by a factor 20. And if we do this, if you look at the experimental plot on the left at the yellow curve, we get, and also at the experimental image, we get a profile that is decreasing very steeply because the GFP, the receptors are so numerous that they capture immediately all GFP that is being produced. And then one gets a very short gradient. Okay, so what do we learn from this analysis? So we get a picture for what the morphogen gradient looks like. And so this is a schematic roughly to scale. There is a thin space, which is about 20 nanometers wide between the cells. There is an incoming flux from the source of 2 molecules per second. Near the source, most receptors are bound. And far from the source, actually, most of the GFP is actually bound and not free. So if we look at the numbers we extract from our analysis, we see that there is about 1,000 times more bound ligands than free ligands. So freely diffusing ligands or morphogens are actually quite rare. They spend most of their time bound to a receptor. And this also allows to figure out one potential issue, which is that if one looks at the formula that gives the length scale as a function of the diffusion constant and the degradation rate, if the diffusion constant is high, which one gets if one takes a value for GFP freely diffusing in water, and the degradation rate is low because the receptors are only internalized one every few hours, we would obtain a very long gradient. In other words, at the scale of the tissue, no gradient at all, the profile would be completely flat. But in fact, what's going on is that if we look at our numbers, a ligand stays unbound for a very short time, less than a second. Then it binds to a receptor, and either it gets absorbed with a 20% chance, or it has 80% chance of being released after 1.5 hours. And so overall, the typical trajectory of a morphogen in the tissue is to stay 1.5 hours, live for less than a second, stay for 1.5 hours, etc., about 5 times before which the morphogen is absorbed. And so that means that effectively, it's only diffusing freely for about 1.5 seconds. And so it cannot go very far, even though it spends a lot of time in the extracellular space, but a large fraction of its time is spent bound to a receptor. Okay, the other thing we can do is see if the gradient is functional. And so, because the gradient has a function, which is to lead to domains of target gene expression. And so each cell sends with its receptor the level of morphogen, responds by a signal, and triggers the expression of target genes. And so this is the French flag model, which assumes that one can generate a pattern in the tissue based on different levels of morphogen in the system. And so, but if we think about this, we see that there is an inherent constraint due to the fact that at steady state, it must be that every produced morphogen in the source must be degraded either in the tissue or in the hemolymph, because it must be that production balance total destruction. And that implies that if one does not degrade enough in the tissue, a lot of the GFP is going to go in the hemolymph. That creates an issue because if we assume a reasonable gradient, which generates a domain where a target gene is on and another domain where it's off, which can then be used to specify the position of the veins, for instance. If one has a low number of receptors in the tissue, one gets a very weak gradient because there is little degradation. And one gets also a high level, a high background level, which can lead to target activation everywhere because of a lot of the morphogen is going in the hemolymph. And conversely, if we try to compensate for this and put more receptors, we can now degrade the GFP in the tissue. It's not going in the hemolymph anymore. There's no background level, and the gradient becomes very short. And so one needs to balance these competing requirements. And this can be summarized by looking at the phase diagram, where on the x-axis there is the receptor density. On the y-axis, there is the lifetime in the hemolymph, how fast the GFP will be degraded in the hemolymph. And there is a region in the diagram where there are not enough receptors, so not enough signal, so no target gene expression. There is also a region where, because of the background level, there is full activation. There is a region where the gradient is very short because there are too many receptors and the degradation is too fast. And so there is only a tiny region in this phase diagram where one can obtain morphogen gradients, which will create reasonable domains of target gene activation. And so to test this, it's actually possible experimentally to genetically modify the receiving cells inside the wing disc so that now the receptors are the real receptors for DPP, but they bind to the extracellular GFP, so only the extracellular domain is changed. And they form dimers because that's how DPP behaves. And then the intracellular part, which is the normal part of intracellular DPP signaling, can trigger the real cascade of signaling inside the cell, which is PMAD activation and expression of target genes. And then, so this is what a normal wing would look like with DPP. The green region shows the level of PMAD, so the molecule that is activated by DPP. And one can measure the gradient of PMAD away from the source. It looks like a decreasing exponential, roughly. And we can measure the size of the target gene, here Spaltz, which is thought to instruct the position of veins. Now, if one does the experiment with a single, with a pair of signaling receptors, and we measure so there is a response, there is a PMAD response, but we see this problem that we observed in the theory, which is that there is a background level which is high because not enough GFP is being degraded in the tissue and it goes in the hemolymph. And then we get target genes that are broadly activated and do not have the normal activation size. If we now increase the number of signaling of receptors by expressing twice as many of them, the PMAD gradient becomes very short and we get rid of the background level. And the target, the domain is too small. So indeed, it's difficult experimentally to find parameters for which we would get the right response. And we can also look at the adult veins that are obtained. So the left vein is the normal one. And already remarkably, even so without DPP, there will not be growth. The artificial morphogen recapitulates growth. But with one signaling receptor, there are too many vein cells, and possibly with twice as many signaling receptors, not as enough vein cells. So the next idea, was to see if we could make the situation a bit more complex and introduce a second non-signaling receptor. So with the idea that it could help with transporting the morphogen, but not signal. And if we just add a fixed non-signaling receptor in the theory, we see that this doesn't help because the non-signaling receptor creates a new way of degrading the morphogen without helping its transport. And so if we compare the blue curve to the orange curve with the addition of the non-signaling receptors is worse. But then if we think of receptors that can diffuse and possibly hop from cell to cell, which is something that has been observed for GPI-anchored receptors, then we can find, if we introduce additional diffusion of the non-signaling receptor, we see that we can simultaneously expand the gradients and get rid of the background because the non-signaling receptor absorbs the morphogen inside the tissue and promotes its transport. And indeed, if we start again from the situation with a signaling receptor, which produced a gradient, but with a high background level, and we had a GPI-anchored non-signaling receptor, we now get a gradient that looks better. It does go to 0 at large distance and is relatively wide. And the domain of target activation looks better. And if we finally look at the wing, we obtain by doing this the best looking wing of different experiments. But overall, what's remarkable is this wing has been patterned by an entirely artificial GFP morphogen. Okay, and so to conclude, I've discussed two situations of where scale is changing in morphogenesis. One in mechanics, where one goes from apical and basal cellular forces to a tissue scale deformation. And another one is signaling, where one tries to relate the molecular properties of how a morphogen binds to receptor to and how these parameters can be tuned to make a functional morphogen gradient, which can be used for patterning of the tissue. And so I would like to acknowledge my group, notably Luca and Marc, who have worked on the second part, and Diana and Sylvanus on the first part, as well as my collaborator, Axel Berens and Jean-Paul Vincent. And would like to mention that I'm recruiting currently postdoc and PhD students. And thanks for your attention.